So today I'm super happy to have Samira Abdulai-Saiku here. She joined faculty at Temple last year. So she's had a lab for about a year. She's going to talk through her journey here, but just briefly, she did undergrad in Ghana. She then did her PhD in Singapore, and then did a postdoc at UCSF. She's been telling me a lot about the research she was doing. She studied toxoplasmosis, which is a fascinating story. I'll let her tell most of it, but it changed a parasite that infects mice and then makes them want to be eaten by cats, basically. A very bad simplification. And then has been out of UCSF doing more work on the effect of sex differences on cognition. Aging. So, take it away.
Thank you. Good afternoon, everyone. I'm glad you made it, and I'm happy to talk to you both about my scientific journey and then the work I have been doing since I've been a postdoc. But I'm no longer a postdoc, but you get the idea. So as Joel mentioned, I did my undergrad in Ghana, the KNUST, which is a university in the Ashanti region of Ghana. And part of our degree, because it's an honors degree, requires that we do a final year project. So it was really the place where I first learned how to do research, to ask questions, collect data, write it up, all those things. And I was in my final year, I worked with Dr. Arthur, who was my project supervisor, and Charles Apple, who's now a professor himself at the same university, who was a master's student at the time. And we were looking at how nutritional status of children affected their response to chemotherapy treatment when they were diagnosed with Burkitt lymphoma and Wilms tumor. I really enjoyed the process of, having a question and asking these interesting questions, gathering the data, having to go to the hospital to gather the data, write about it, present my work to the professors and my colleagues, and basically do a defense where they tried to tear apart my work, and I needed to really understand what the work was about to answer the question. What I did not enjoy was seeing the children in pain and all that. So I realized maybe human research was not the thing for me. But after my undergrad education, I worked for about a year at the Noguchi Memorial Institute for Medical Research, which is a research institute attached to a different university in Ghana, which is closer to where I lived growing up. So it was easy because I went back home and I could work here. Ghana has a very interesting national service program where rather than going to the military like many countries do, you get put into the workforce to work for the government basically for a year. And this is because our college is heavily subsidized, so we kind of work for a year to pay it back. And when I was here, I worked on parasites, just all kinds of parasites. I had some introduction to Noguchi prior because I had been interning here for the summer since the end of my second year in undergrad. But in my once I was done with college and I worked here for about 15 months or so, I worked with Prof. Wilson and Prof. Anita Gansa, and we worked on mostly every parasite you could think of. But I really spent a lot of time working with malaria. So this is why I learned how to culture malarias, identify the stages of malaria, synchronize the parasites to specific stages for research and all those things. But right around the time that my time here started to end, we started to think about working with a parasite known as Toxoplasma gondii. And so it was in the very early stages, but I'd already started thinking about what was next and grad school. So I was in the process of applying to grad schools. And that is when I came across the profile of Dr. JVS, who was working on Toxoplasma and how this parasite could basically change the way rodents viewed a predator from completely avoiding the predator to being more willing to explore areas with predator stress. around it. I thought it was really interesting. I had initially thought I could just get away with doing some biochemistry for my PhD, and work on the mechanisms underlying this attraction, not on the predator versus prey side, but also because toxo was involved in increasing attraction to infected animals, which almost never happened, trying to understand that. But that didn't work out because somebody else was already doing that project when I got there. So I had to suddenly learn what the brain did in a way I hadn't thought about ever, because as somebody who was a biochem major in college, it just kind of never came up. Um, so I had to learn a bit about the brain regions and what they did and how this parasite was affecting the way the rodents perceived predator odor. But what was interesting to me is that every mechanism that the lab had worked on, that other people have been working on for the last 20 years or so, was very male centric. It involved testosterone, it involved medial amygdala, AVP expression. It was just things that were not very common to females. So I walked into Ajay's office one day and I said, "What happens in females?" And he said, "We don't know." So he said, "Do you want to find out?" And I was like, "Sure." So I did the experiment and I found out that the same loss of fear to predator stress happens in females also. The problem was that we couldn't get any of the mechanisms that the lab had been working on for ages to show up in the females, which told us that something different was happening. So I went down this rabbit hole of figuring out that there was no testosterone, no estrogen, no progesterone, none of the typical circulatory hormones involved. It was oxytocin rather than AVP in the brain. And it just seemed to be a completely different mechanism driving the same phenotype, which was a little weird, but it also kind of got me into the space where I became really interested in sex differences, both sex differences that happen phenotypically, but those that happen mechanistically. So as I was done with my grad school, I was thinking about what was next. I figured I still enjoyed research, so I wanted to do something sex differences related, but I didn't know what. And then I think I cold emailed Dr. Dina Duval, who eventually became my postdoc supervisor. She reached out to me and we started talking about the possibility of joining her lab. And she, on her website, had interesting things with hormones and stuff, which is what everybody thought of sex differences being, like hormonal differences driving the difference in phenotype or mechanism. And then I think in our second interview, she said, "Yeah, well, but we want to look at how X chromosomes are driving sex differences." And again, I had the same like moment where, wait, sex chromosomes do something except denote as male and female. That was completely news to me. And so we started doing that work, and it took us down several rabbit holes. The figure in the middle is a transgenic mouse that has fluorescent X chromosomes. So you will see me show it a couple more times as I talk about the work that I did in the lab. But basically, my scientific journey has spanned 3 countries in very different parts of the world, and also has spanned very different scientific interests. I started in parasites, and then I did something in the brain, and then I did some aging, and then I've done some cognition. And I think the consistent thing for me now is that I think my love of science centers around my love of sex differences. And I'm not really very rigid about where that takes me. I just want to understand sex differences. So before I talk about my work, is there anything you'd like to clarify with my scientific journey? Great. Okay, so today I'm going to talk about how the parent of X origin mediates cognitive performance across the lifespan. So female mammalian cells have two X chromosomes. One they inherit from their mom and the other they inherit from their dad, whereas males only have a single X chromosome. So there's a need to normalize X gene expression between males and females. So females have evolved to um, use X chromosome inactivation. This is just a random and independent process where each cell in the female silences one of the two X chromosomes. And this makes female mammals mosaics with some maternal X expressing cells and some paternal X expressing cells. This is one of my favorite pictures. It's also one of the only visible forms of X chromosome inactivation in nature. And it occurs in calico cats. So what you see here are twin calico cats, and they show different patterns of a red and black allele of an X-linked coat color marker. This is the hippocampus of a female mouse. Here, the cells you see that fluoresce green express mom's X, and the cells that fluoresce red express dad's X. What you see here is a mosaic pattern of red and green expressing cells, and it's believed this mosaicism is able to buffer dysregulation that occurs in aging and disease. So there are three main X chromosome mechanisms. We've already just talked about the X mosaicism because females have some cells expressing mom's X and some cells expressing dad's X. If there's a mutation on one of these X's, you have a second population of X expressing cells that are able to buffer these adverse effects from the mutation and use. So this is why usually X-linked diseases have much less symptoms in females than they do in males because males don't have any other buffer. X chromosome escapees. Remember when I said one of the X's are silenced? That is true to some extent. Even though it's supposed to be silenced, there are about 3 to 7% of genes that escape silencing. And so very originally, we refer to them as X chromosome escapees. And these have also been shown to contribute to resilience in female mouse. Today, I want to talk about the parent of X origin. And it's kind of an extension of X mosaicism, where the females have a second X. But my argument here is that it matters which of the X's is actually active, because it's not just having an X. Just having an X is valuable, but what X is even more valuable than just having any X present. So why would we want to study the parent of X? Why is it important? Well, we know from studies of women with Turner syndrome that the parent of X is actually very important for cognitive performance. So individuals with Turner syndrome are individuals who only have a single X. They do not have a second sex chromosome. So no second X and no Y. Because of this, they are phenotypically female. These individuals show cognitive deficits when they are compared to females with two X's. However, in a very intriguing study where they tested these females against each other, so they grouped them into those who get their single X from their mom and those who get their single X from their dad. And when they compare their cognitive performance, they realize that those that get their single X from their mom perform worse cognitively than those that get their single X from their dad. And so then it made us wonder that could the parent of X be important for driving cognitive performance outside of the disease model that is Turner syndrome? Additionally, because they only have a single X, they lose mosaicism and then they lose SSKP expression. And so either one of those, especially SSKP expression, could be driving whatever phenotype was seen. So then we wanted to do things a little different. We're going to keep the second X, but we're going to skew X inactivation. So we created a transgenic mouse model where the mice still had two X's, but rather than allow them to allow each cell to choose, like it happens in wild type, which X they silence. We forced the cells to silence dad's X and only express mom's X in every cell of the body. And then we characterize these mice across the lifespan. So first, we wanted to see how skewing of the maternal X affected cognition in young female mice. We found that maternal X impairs spatial memory in the young female mice. We tested spatial learning and memory using the Morris-Water maze. So for all of the graphs from these transgenic mice, the wild-type mice are going to be denoted XMXP. The maternal X-only mice are going to be the XM. And here you have latency to find the hidden platform and number of testing days. And you can see consistent, similar learning across all testing days for both mice. But then when we probed their memory of the platform location 24 and 48 hours after the last day of testing, we found out that the maternal X consistently showed impaired memory of the platform location at both time points. So then we wanted to know how skewing of the active X affected key physiological functions in middle-aged female mice. Remember that this modification was whole body, not just the brain. So we're interested in how it affected everything else. We chose middle-age time point for this testing because that's usually when an adult you begin to see deficiencies arise in humans. Interestingly, we found no differences between the maternal X and wild type mice in heart function, bone density, body composition, or in metabolism. Everything was pretty much the same. So we concluded that it wasn't really doing much outside of cognitive performance. But just to be sure, we aged the mice up. Excuse me. And then we asked what happened when we tested cognitive performance in these aging female mice. We found that the maternal X worsens an index of forgetting in middle and old age. So here we use a well-established paradigm of repeated testing in the open field to measure memory. The test measures the ability of the mice to remember the spatial context of the open field by comparing their activity levels on the first day of testing in middle and old age to their activity levels of the last day of testing at the previous time point. And here you have index of forgetting on the y-axis and testing stage on the x-axis, and you find that the maternal X mice show increased forgetting compared to wild type at middle age, and this worsens in old age. But I think what is pretty striking here is that old wild type mice are only as forgetful as middle-aged maternal X mice, which told us that something about maternal X only expression was accelerating cognitive impairment in these mice. So then we began to wonder whether this could be due to accelerated epigenetic aging. So your chronological age is how many years you've been on the earth. Your epigenetic age is how well your cells are functioning at any given time. So you could be chronologically 30 years old with cells that are epigenetically 15 years old because you take really good care of yourself, or 60-year-old because you take really terrible care of yourself. And the way we do this is that we harvest tissue from these mice, we extract DNA, and then we look at the methylation patterns at age-associated loci to predict their epigenetic ages. Now, the first thing we did was blood cells. And interestingly, we saw no difference between young and old mice in the epigenetic age of their blood cells. Now, this could have equally been the chronological age graph because this is how similar they are. So keep that in mind for the second one. So then we looked in the hippocampus and it was strikingly different. So again, the young mice had no difference in their epigenetic age, but their old mice showed that the maternal X mice had increased epigenetic age compared to the wild type mice, which means their cells were aging faster than that of the wild type mice. Now, I don't want to say we didn't believe it, but we were really struck by the observation. So we decided to repeat the experiment a different way. We took these transgenic mice we had. These are fluorescent tagged. These are mice with fluorescent tagged X chromosome. So in our model, mom's X fluoresces green and dad's X fluoresces red. This allows us to use fluorescence activated cell sorting to separate these two populations of cells and then do the same epigenetic age analysis. And it is important to keep in mind that these are B6, wild type B6 mice. So they are congenic, they have zero genetic variability within them. And we see pretty much identical data. There's no difference in young, but very striking difference where maternal X expressing neurons are epigenetically older than paternal X expressing neurons. This is also striking because these samples were collected from the same brain. These are samples that have lived side by side in the brain, experienced the same environment and everything. So we knew that something intrinsic was causing these differences and could not be simply attributed to things that had happened in the mice across their lifespan. But because epigenetic age is measured by looking at methylation patterns, and methylation patterns were different, we wanted to see if this was affecting gene expression in any way. So we used the same mice. It allowed us to sort them out and do bulk RNA-seq. Now, I should preface this by saying we expected to see minimal changes, some changes, but minimal changes, maybe 50 genes, something. We saw 2,000 genes changing across the genome, with about 80 of them being X chromosome genes. This is a PCA plot that basically condenses the data into 2 dimensions so you can view it all at once. And the greater the separation, the more difference the gene expression between the 2 populations are. As you can see, there's no overlap anywhere. They're really, really separate. This is for the young, I believe. And we did the same thing for the old mice also, and the PCA is about 80%. PC1 is about 80% for that. So we had about 2,000 genes changing across the genome. But what was really interesting to us were these genes that showed an almost on and off expression pattern within the genome. And these are all X chromosome genes. So people who had studied Turner syndrome in mice had seen a similar on and off, but in their study, they had done a whole brain. This was just neurons. They'd done a whole brain and they'd identified XLR3B, which showed really high expression of mom's X with almost no expression of dad's X, which we also see in our data set. I'm just not showing it now. But in addition to that, we were also seeing expression of about 9 genes that were showing really high expression of dad's X with almost no expression of mom's X. And of the 9 genes, these were the ones that were the most robust and the most consistent when you looked at the values across. So we began to wonder if maybe one or more of these genes could be modulated in aging mice to improve cognition. But then we didn't know which of the one because, spoiler, X genes are not really well studied, so we don't know a ton about what they do, especially cognitively. So there was no literature anywhere to tell us possibly if these could be contributing to cognition in any way. So rather than choose and get it wrong, we decided to do all three. And we did we use CRISPR activation to achieve this. So this is the CRISPR activation plasmid that is doing the heavy lifting. This is a guide RNA that is not targeted to any gene and is going to be our control. And this is the guide RNA plasmid that is targeted to TLR7, SASH3, and CSL1, which are our genes of interest. So we did bilateral hippocampal injections, specifically into the dentate gyres in 18-month aging female mice. And then we tested their cognitive performance. So when we increased expression of our three genes of interest, we saw increased memory in the Morris water maze. And we also saw increased memory in what is known as the novel place preference test. The way that test works is that if mice are more likely to explore new things than things they already know. So you put them into the environment, new things and new areas. You put them into the environment, you give them two objects, whatever objects you choose, but two identical objects. And then they explore it for about 5 to 10 minutes. And then you take them out. For us, we use a waiting period of 4 hours. And then before you put them back in, you move one of the stuff. If they have a good memory, they're going to know that thing has moved and they're going to spend more time exploring that. And that's a very hippocampal dependent memory. If they don't have really great memory, they're not going to discriminate between exploring either one of them. You can see here a discrimination. This is a discrimination index where it's just time spent exploring one object versus the other, and the newer object versus the one they already knew. And you see that the control old mice have a discrimination index close to 1, which means they couldn't really tell the difference. They didn't have any memory to know this was a new one. But these guys performed better compared to the control mouse. So we basically come full circle, where we started with a question about how the maternal X affects cognitive performance in wild-type females. We were able to show that skewing towards the maternal X was harmful for cognitive performance, particularly as you began to age, and it even accelerated aging. We showed that it also accelerated epigenetic age of hippocampal and cortical neurons, and that it drastically affected gene expression. And we were able to identify 3 genes that, when modulated together, improved cognition in aging female mice. And so what this tells us is that our ability to understand how the parent of X regulates gene expression and, to that extent, cognitive function in females could yield important mechanisms and pathways that could benefit both males and females. And that's kind of the work I am doing now. It's taking some of the genes we saw in our screen, comparing maternal X and paternal X expressing neurons, and then seeing how they affect cognition, not just in females, aging females, but also in aging males. Yes?
So you said the 2 X chromosomes are genetically identical.
Genetically.
But somehow there's a huge difference. What's the difference between the two? I mean, you're fluorescent tagging them, but in the actual animal, when you're not fluorescent tagging, what's different?
No, they're B6. There's absolutely nothing genetically different. We think the difference is epigenetics. We think the difference is in how the cells are inherently marked. And we know they're inherently marked a little differently because we go back into development when X chromosome inactivation is occurring, particularly in mouse studies. There's a period where the mouse has to specifically inactivate that X. And if it makes a mistake, the placenta doesn't form and the mice die in utero, right? So we know the cell can tell the difference, which means they are marked differently in some way. We just don't know how, what the differences are.
And then you had 3 genes in there, but I think I missed the step of how you pick those specific 3 genes.
Oh, so we have 9 genes that show that basic. I'm only showing the 3 we picked here, but we have 9 genes that show this kind of pattern of expression. But of the 9, these were the most robust. So we had 5 samples each in our RNA-seq comparison, and when you looked at them, these ones just showed the consistently always high in XP, XM, just huge differences. So we stuck with those. There's a little bit more variability in the others. Yes, that's all I got. And all these people who have been funding my work and helping me with my work, I don't know what I would have done without Dan, who kept the colony going, and that's how I survived my PhD. Do you know who was my advisor for all 7 years of it? And Barbara and Sandeep, who contributed both mice and expertise to that. And Simons, who funds me, and NIH and AFA that funded my PA. Happy to take more questions. Please, comments, disagreements. I'll take all of them. Confusions. That's an important question. We're trying. The problem is, so remember when I said there's a point of time when in development in utero where, and this is really early on, where the cell specifically has to shut down that X expression. If it doesn't shut down that X expression, the placenta doesn't form. So the problem is once the placenta forms, you have a really short window before random X inactivation happens. So you're going to have to time the forced silencing between placenta formation and random X inactivation happening. And finding a good, we use a Cree-based system. Do you understand what Cree-logs is? Somehow. Okay, let's go. Let's kind of travel back in time. Okay. Here. So on the maternal X, you can see a little delta exist over here. We deleted the exist, but because there's no point in the developmental process of the embryo that the maternal X has to forcibly be shut down for development to occur, we're able to do this whilst it was still an oocyte before fertilization. So we didn't have to worry about a time window. But because we can't do it in sperms because they need to be shut down later, and if we do the deletion, we are now restricted to a specific time window. And getting a good Cree that can do effective deletion because the exist is fluxed, right? So we can use a Cree to delete it. Getting a good Cree has proven really difficult. We've tried. So we are still trying. We are still like figuring out Crees. We are basically doing a process of elimination right now. We have to target it specifically because unfortunately after placenta forms, the cells that form the placenta still need to remain silent for the entire duration of the pregnancy. So we need to stick within the epiblast, and that has been slightly more challenging than we would like. But we've received some other methods by which people have suggested we go about them, and it's part of the things I'm working on in my new lab now.
So you're saying that the mice were the ones that they're very like genetically
Identical?
right?
Yeah.
So how are you seeing any difference? Because if course people like they are genetically the same. So how are you tracking the maternal? Where are we going? How do you know which one is which?
How do we know which gene is maternal?
Versus paternal, if they're all the same, if you don't have them.
Okay, so to get this mouse, we are breeding two mice, right?
Yeah.
So we know that the male coming in is a wild type. He's a regular male, there's nothing wrong. We've not deleted anything. So he's coming in with a normal X. But the female coming in has this deletion already. We do this deletion before the mating. So we already know she has this deletion, right? The eggs she's producing, it's a heterozygous. So the eggs, some of them are normal and some have the deletion. So when they're born, we do a genotyping, right? We do a PCR test. We take a tiny bit of their tail because it's a whole body manipulation. It doesn't matter which body part we take. We take a tiny bit of their tail and then we genotype it. And based on the size of the bands, we know whether we're dealing with a wild type or with a maternal only expressing.
So you talked about cognition, which I think all came from that early finding in the humans with the maternal. But do you think this is really broad and affecting many different things? Or do you think it's specifically cognitive for some reason?
I think we've not seen, we didn't see it in our data set when we looked at physiological function. So it clearly didn't change anything for us. But there are caveats to that. One of that is maybe we tested too early in middle age, and if we'd waited until they were really old, we'll see something. So that's that. I also happen to know there's a study in the immune system of parental X. They use something similar to the Turner syndrome model where they chuck an X out and they look at how the parental X affects immune system function. I believe they use two specific autoimmune diseases. And it turns out in your immune system, having that X is terrible for you, right? Which kind of makes sense because if Mumps was universally bad, it would have evolved out of the system by now, right? Something would have chucked it out somewhere. But it's not. It seems to be very tissue and organ system specific. And in the brain, having Mumps is bad for you. But it turns out in the immune system, having Mumps is actually beneficial for you. So we might have tested too early in our physiological functions and just not caught it. But we didn't see any like gross weight differences and we tracked them. So I don't know how I feel about that. But yeah, it might be that it could also be more subtle than whatever we tested for. So there are those caveats. Yeah.
More transition to human life with more cognitive degeneration.
Gains? Entirely possible. The problem with the brain is that we can't just go in and randomly just turn everything on or turn everything off, right? That would be catastrophic. So we're going to have to really understand how to specifically fine tune. Like the data I showed you here, right? We did this specifically in neurons. We used this synapsin promoter to make sure that the changes will occur only in neurons. Because SHTI, TLR7, and cis-LTR1 all have immune functions and can be pro-inflammatory, right? So if we just turn it on anywhere, that's going to be a problem. So we're going to have to figure those kind of specific things out. Like, what do the genes we are trying to turn on and off do? Is it something we can get away with, like global expression? How targeted does it have to be? Is it something that can be put in the blood and will work once it crosses the blood-brain barrier? Can it cross the blood-brain barrier? There's a lot to be figured out, but I think between these and like human data sets, people are doing longitudinal tracking of aging individuals. We have just regular tracking of normal aging individuals where people join a study and for years and years and years, they get checked every 6 months or every year. And usually when they die, these people donate their brains. And so we're beginning now for a lot of these studies that started in the 80s and 90s to have tissue and be able to compare gene expression from humans to mice and see if things track the way we think they do. And because they are tracked, the humans are tracked, we can tell those that develop dementia what their gene expression changed to look like, those that did not, those that just had regular aging induced, memory decline. And we've also started these, not us in the lab, but as a scientific community, have started these studies where we have individuals we call super agers who are more than 100 years old. They're usually centenarians who don't show any cognitive impairments. And they're doing really well for themselves. And we're tracking these people. So hopefully they donate their brains when they die. And then we can figure out what makes them stay cognitively intact. So I think we'll get there. It will take longer than a systemic disease, but I think we'll get there.
Leads to worse cognitive outcomes in female mice. Does that also lead to worse outcomes in male mice?
It does. It does. They perform worse in many aspects of cognitive performance, particularly during aging than female mice do. Yeah.
But you were like testing memory. Did you test this for chip memory or or is it like a
So there is a difference. So when we do this, which is this one, which is just 4 hours away, that's a type of short-term memory testing, right? But when we do that's long-term memory, because we start them when they are young and we continue to test their memory of the space until they age. The time difference between this last day of testing here, and the first day of testing here is about a year, right? So that is long-term memory. And this is from here to here is a few months. So we did both long-term and short-term memory, and yeah, still the maternal X mice were impaired.
Great. Well, thank you so much for coming.
Thank you. Appreciate your time.